हेलो गाइस एंड वेलकम बैक टू एक्सेलशियर एकेडमी। मैं हूं आपकी डॉक्टर रूबी सिंह एंड आज मैं लेने जा रही हूं लेक्चर 5 ऑन प्लांट टिश्यू कल्चर. सो टुडे व्हाट वी आर गोना स्टडी इज डिफरेंट टाइप्स ऑफ प्लांट टिश्यू कल्चर डिपेंडिंग अपॉन द मीडियम. ओके, नाउ व्हाट डज इट मीन? सो whether it depends on the like solid media, semi-solid media or suspension media. So depending on that, we have got different types of culture and depending on the part used for the culture. So what does it mean over here? It means whether you're going to use the leaf or flower or nodes or internodes or the meristematic tissue. So different parts of the plant, whatever you use, depends on that. You have got different types of culture. Okay. So first what we are going to talk about is depending on the type of the medium used. So we have got different types. First one is the suspension culture. So suspension culture, by the name itself, we know that over here they are talking about the liquid one. So these medias are remaining in the liquid phase. Okay, so the callus or the plant cells, they will be suspended in the liquid medium. Then we have got the static culture. So that is, you have got the agar base over there onto which the callus is being inoculated and grown further. Then this suspension culture is again divided into batch culture, semi-continuous and continuous culture. Then again under the the continuous culture, we have got open and closed type. So again open, we have turbidocytes and chemocytes. So sit tight because right now we are going to talk about all these different types of culture in detail. Okay. So yes. So first one, what we are going to discuss is the static culture. So from the name itself, it's clear. This is stationary. Okay. यहाँ पे ये सब रुका हुआ है. It is not moving. So what you do is you inoculate the media and keep it in the incubator. बस और कुछ नहीं करना होता है इसमें. So okay. So callus, already we know what does it mean. It means that it is the mass of undifferentiated plant cells. ठीक है. So what happens in this? So whatever part you have used of the plant, it actually gets de-differentiated. Okay, it gets de-differentiated. That is, it goes back to the condition into which it is not, you cannot say that the plant ka hai, sorry, you cannot say that it is the part of the shoot or root or leaf. Okay, so that is the de-differentiated. Now, so this is the mass of undifferentiated plant cells which can be grown on the solid media from the any part of the explant. Okay. Now, it may initiate from explants of any multicellular plant. So, any plant can be cultured in the plant tissue culture. It just that unke requirements alag-alag hoti hai. Okay. Now, the organs which can be used are root, stem, leaves, flowers, fruits, etc. Now the cells are initiated from the explant or the segments of the root, stem or leaf, either from the mature or the embryogenic plant. So you can also use the embryogenic stage of the plant in order to carry out the plant tissue culture. So it is actually depending on your interest कि आपको किस तरह की study करनी है. So depending on that you actually decide कि आपको mature plant लेना है या embryogenic stage पे लेना है. Then this explant excise fragment of parenchyma or mixed tissue containing a cameo or endosperm. Okay, so again, depending on what you have selected. So the longer the tissue explant, the more complex the range of cell types and greater the possibilities of initiating a culture of mixed cells. So what does it mean over here? It means that if you take this much big fragment, so the chances of having mixed cells is high in this as compared to this much small fragment if you take. Okay, so if you take small piece, then the chances that it has got 100% same type of cells are more as compared to the chances of having such a big fragment. So whenever we make or whenever we start plant tissue culture, what we do is actually we make the small pieces and each small piece will be then inoculated into the media. Okay, so the callus can be induced to under organogenesis. Now, what does it mean organogenesis? That is the synthesis of new organs, and embryogenesis, and eventually the whole plant by providing suitable nutrient media. So don't worry, with the further coming lectures, we will be talking about organogenesis as well as the embryogenesis too. Now, what will be the advantages of using this static culture? You can study the biosynthetic pathway using tracer elements. Yes. So all the pathways which are carried out, like for example, the regulation by the growth factors, phytohormones like auxin, cytokinin. Many studies have been done usually using this PTC and they have determined the biosynthetic pathway. Okay? So when we are going to discuss about the plant physiology, so there we talk about different pathways for the growth factors. So there, this analogy, that so there we use this technique. Okay, then it is very much helpful in the metabolite production, secondary metabolite production. So this is also very useful because most of the secondary metabolites produced by the plants, they act as the antibiotics, or mainly the anti-cancer drugs also are being made. Okay, yeah. So next, so this is just the pictorial representation how you can actually carry out the static culture. So tissue from the sample from any region of the adult plant can be used. So already here it is mentioned that we are using the adult plant, so it means we are using the mature plant. Okay, over here. Now, so you have got the plant, you can take the bud, leaf, or stem or root, any part you can take. So whatever you take, you are going to take the smallest part and put into the media. Now this is your, let us say this is your MS agar media, solid Murashi scoop media. Now, with the period of 4 to 8 days, this plant tissue will get de-differentiated or undifferentiated and callus is formed. Okay. Now, what you can do is this callus can be broken down into small pieces. Okay. Either by agitation or with the mechanical pressure, you can just break down into these into small pieces. Now, each piece now present in the suspension media. Now this is in the suspension media. Even with the help of the agitation, you can break down this callus into small pieces. Now this small piece will be inoculated into the solid media, from where, depending on the type of the hormones you provide. So when you need the entire plant, see, you can see over here the entire plant is there, right? So you have got the shoot, as well as over here you can see the roots are also present. Okay, so you will give a very good proportion of both auxin as well as cytokinin. Okay, so that's how you can get the whole new plant. So this is how your static culture, the callus culture, is carried out. Now talking about the suspension culture, so it involves active proliferation of callus as undivided units suspended or submerged in the liquid media. So you can see over here, the conical flask is there and the media is present. So you can have different media also. You can see the difference in the color also. Please label it whenever you work in the plant tissue culture. Whenever you do any research, you are supposed to label it. Otherwise, the next day you won't remember what samples are present in which culture. Okay, that's why labeling is very important. Now you can see they are kept on the shaker. So you can insert your conical flask over here, and with the help of these with these regulators, you can actually, control the speed of the shakering. So usually we keep around 150 to 200 RPM. Now constant agitation is required. So it will prevent the cells from getting settled, or it will also prevent it from becoming the clumps. Okay. Then the suspension cultures are initiated. This is R, not A. It is initiated by transferring the pieces of these undifferentiated callus into liquid media. And then again, it is agitated during the incubation. So even though we are saying that it is getting incubated also, actually, if you have this kind of shaker, so then the entire room is controlled of having the same temperature and humidity and light. Okay. Otherwise, they have got the separate incubator boxes there. So under that, you can keep. Now, batch suspension culture. So you can see that slowly we are talking about different types of the culture we are using depending on the type of media we use. Now, batch suspension culture. So what does it mean? It means you are going to culture in a fixed volume of a medium. Again, iska kya matlab hai? It means that if suppose I have started with 1000 ml of media, I am going to use only that till the entire experiment is completed. Okay. So that means in that particular batch, one slot of plant tissue culture has been completed. Now, in batch cultures, as the cells grow, the medium is depleted of nutrient and metabolic byproducts from the cells accumulate, right? So this is again like the growth curve. So initially it will take time, that is your lag phase. Then it will start growing, okay? So that is the exponential phase. And finally, your secondary phase will come, stationary phase will come. So during that, you can see that the metabolic byproducts, that is your secondary metabolites, will be produced, and the medium having the nutrients will get started, it will start depleting. Okay, very much similar to the bacterial growth curve. Now these batch cultures, it can be characterized by continuous changes in the medium, okay, continuous internal changes in the cellular composition, and accumulation of metabolic products. So all these tests you can do in order to, in after, like suppose you started with 1000 ml of the media, then 0 are what are the composition, then 12 hour is what you're going to get, then 24 hours what you're going to get. Okay, so depending on that you can study all this. So the system is closed with respect to additions or removal of culture except for the circulation of air. So what does it mean? आपने इस batch culture में extra medium नहीं add किया है and कुछ भी remove नहीं किया है. So if I have started with 1000 ml, so it means throughout my experiment only 1000 ml of media was used. Okay. Now talking about semi-continuous suspension culture. So this is open system. So what does it mean? There is a periodic removal of culture and the addition of fresh medium. तो इससे क्या होगा? The growth of the culture is continuously, it is maintained. ये रुकेगा नहीं. Now in the open system, in this the volume of culture remains constant, but हम इसमें fresh medium add करते हैं and पुराना medium निकाल देते हैं, so that there is no accumulation of the byproducts and cell proliferation will be under constant condition. तो इसमें ये grow होता रहेगा. Now this system allows establishment of steady states of growth and metabolism. This is also important. Please remember. Then study the changes which occur in transitions from one steady state to another, and you can identify the controlling factor. So what you can do is certain factors or certain parameters you can change by keeping other parameters constant, and you can study the changes happening in the growth of these cells. Okay. Now, as we were talking about, there are two types of continuous suspension culture, open and closed. And under open, we have got the chemostat and terbidostat. Ab iska kya matlab hai? So open type means, again, you have got controlled or regulated way of addition of new media and balancing the harvest of equal volume of culture. Now, in the chemostat, continuous new medium is added at the predetermined rate and determine the nature of resulting equilibrium. So whatever you're doing is, like, for example, you have started and every 12th hour you're adding new media and checking reaching till the equilibrium. So this will help in studying the steady state equilibrium of the culture, and the desired rate is also maintained by adjusting the level of concentration of the nutrient by the constant inflow of medium. So as I said, so you have started with the experiment. Okay, so let us say that you started with 0 hours, and every 12th hour you are adding new medium. All right, so like this, you keep on adding the different same medium, but at the constant inflow of medium will be every time will be provided. Now, what is happening with terbidostat? So in this, the cell density is the main factor. Now that will be predetermined level. So you are going to check the density of the medium using the optical properties of the culture. And if suppose you're removing the previous medium, you're going to add the new medium and again check the cell density. So it will be helping the particular variable for work at the low cell densities. Now, it offers a simplified system for study of effects of physical factors and growth regulating substance in growth rate and associated metabolism. Okay. Now, this is the last one, the closed type. So what is happening in this? The used medium is replaced with the fresh. Hence, the cells from used medium are mechanically separated and added back to the culture. Thus, what is happening? The cell biomass keeps increasing. Okay. So, yeah. So that was all about the type of plant tissue culture depending upon the type of media we are using. So I hope this is clear to everyone, all of you. And if any doubts are there, please don't hesitate to ask anything. You can comment me in the section, or you can also mail me to excelsioracademy1228@gmail.com. So sit tight because many more lectures are there in order to complete the syllabus. Okay. So thank you guys for listening and for your support.